The overall goal of this procedure is to illustrate how to manually trace the human medial temporal lobe structures on high-resolution magnetic resonance images. This is accomplished by first identifying the amygdala in the brain and tracing it systematically from the anterior to the posterior borders. The second step is to locate the longitudinal extent and the general borders of the hippocampus, and then trace it slice by slice in the anterior-posterior direction. Next, trace the anterior structures of the parahippocampal gyrus, namely the entorhinal and perirhinal cortices, in parallel, to better accommodate variations in their mutual borders. The final step is to trace the posterior segment of the parahippocampal gyrus, namely the parahippocampal cortex proper, in the same anterior-posterior fashion. Ultimately, this manual tracing procedure can be used to obtain a 3D rendering and volumetric statistics of all medial temporal lobe structures.
The main advantage of this tracing protocol over existing protocols, as well as automatic segmentation algorithms, is that it identifies all of the regions of the medial temporal lobe relative to their neighboring structures. This can increase localization accuracy in manual tracing, as well as help in evaluating and correcting, if necessary, the automatic segmentation results in a region of the brain that can have considerable variability.
The current protocol is useful not only for anatomical investigations in the medial temporal lobe, but can also be applied to data analysis from other imaging modalities. For example, the functional magnetic resonance imaging to identify functional regions of interest with high accuracy.